Okay, welcome guys. Welcome to our workshop, and thanks for logging in. I know for some of you it's late in the night, so thank you for logging in. I'm Suvarna Nadendla. I'm a lead bioinformatic analyst here at Institute for Genome Sciences at University of Maryland. I'm like this office is about 35 miles from where Jeet and Raja are. So today I'm going to talk about a Common Fund data ecosystem. In short, it's called a CFDE. I'm going to give you a broad overview of a CFDE project and the integration and data harmonization work that is being carried out with CFDE. Okay. Oops, going for, all right. So here are the contents of this presentation. I'll talk about, I'll briefly touch on all these things, not go into super details, but the first one would be the Common Fund and the Common Fund programs, challenges of big data and what is fair, what is CFDE and the CrossCut metadata model, control vocabularies and ontologies, and finally, I'm going to show you a CFDE partnership tool. All right. Okay. Okay. So this is a busy slide here. Before I jump into the details of the slide, I want to just briefly talk about a few terms that you see on the slide. The first one is NIH. Some of you know what NIH is, but for others who do not know, it's called National Institutes of Health. It's a government agency which was founded in 1880 for conducting biomedical and public health research. So it conducts research internally, but also funds external institutes and facilities. Common Fund is a component of NIH, which provides funds, and it was started in 2004. It supports bold programs, scientific programs that catalyze discovery across all biomedical and behavioral research. So this the programs funded by a Common Fund have to meet 5 criteria, and those 5 criteria are that you see on the slide here. They need to be transformative, which means that it must have high potential to dramatically affect biomedical or behavioral research over the next decade. They have to be catalytic, which means that must achieve a defined set of high impact goals within a defined period of time. They have to be synergistic, which means outcomes must synergistically promote and advance individual missions of NIH ICs to benefit health. And they should be cross-cutting, which means the program areas must cut across missions of multiple NIH ICs. Again, IC is institutes. I think it's called institutional centers, I think. Be relevant to multiple diseases or conditions and be sufficiently complex to require a coordinated and trans-NIH approach. And finally, they have to be unique, which means must be something no other entity is likely or able to do. So all these Common Fund programs are funded for 10 years, not more than 10 years. And if you would like to know more about Common Fund programs, here is the link. Right. So in this slide, you are, again, it's a busy slide. These are the Common Fund programs that are available or that are right now active. 1 or 2 are sunsetted, which means that they have completed their 10 years course. So most of them that you see here are active right now. So before I go into the details, so all these programs generate or have generated high value data sets from different omics studies like genomic, expression, proteomic, metagenomic, and imaging studies. So they have generated huge volumes of data. Okay. Okay, there are various challenges with such high value data sets generated by these data centers. So I might say DCCs every now and then. DCC stands for data coordinating centers or the data generating centers. All the ones that you have seen in the previous slide, they're all the data generating centers or DCCs. So you might hear me saying that in future slides. So yeah, there are a few challenges with big data that has been generated by these data centers. Each data center produces different modalities of data, which means that the data sets contain different file formats, different data types, and generated using different techniques. And the value of these data sets is significantly reduced if other scientists are not able to use them. So significant utility comes only from the ability to do mining and analysis across data sets. So which means that the data has to be fair. But what is fair? So it stands for, FAIR stands for findability, findable, accessible, interoperable, and reusable. And I'm going to tell more about it in the next slide. Oops, sorry. Okay. Again, the FAIR guiding principles are defined in the link below here. If you are interested in knowing the principles, you can go access the page or the publication there. So FAIR principles apply to metadata and data both. They describe how a data asset can be made easily findable by humans and also computers. Accessible with user authentication and authorization. So if I have to give you an example what authentication and authorization are, you can say that when you go to an airport, you show your ID, which means that you're being authenticated for your ID at the airport entry. Then when you go for boarding, and there again, you show your boarding pass, which means that you are being authorized to board the flight. So these are two different things, and both are important. And the data should also be able to integrate with other data that is available with other DCCs. And it should also be able to be interoperable in a way that the other data center is able to use, analyze, and process the data. Finally, the data or metadata should be well-defined so that it can be reusable. So all these principles are very well written in this publication. So I strongly suggest you to read that. All right. So here, it's a picture that I'm trying to explain why it's important to have something that is called CFDE. So previously I mentioned there are a couple of data centers under Common Fund. And all these Common Fund programs are generating high-value data sets, as I mentioned before. And each center has its own data model and using different control vocabularies and ontologies to host their data sets. They have their own portal for search and discovery of data, and they have their own APIs. And also, so all the development is happening kind of siloed. And for an external user who wants to search for data, it is difficult for that person because he has to go to each and every data center and access their web portal to find the data of interest. So it's like a lot of effort. It takes a lot of time, and sometimes it's confusing for an external user. Okay, so that's where CFDE comes into play. As mentioned before, CFDE stands for Common Fund Data Ecosystem. It is NIH-funded project, NIH Common Fund project, I should say. It serves as a metadata hub or repository, hosting metadata about the data assets generated at each DCC, which will allow the common fund, which will allow cross-program searches and make discovery of data easy. So the Common Fund data ecosystem is an umbrella effort meant to coordinate across the Common Fund programs to help make their data findable, accessible, interoperable, and reusable. So I'm going to explain more about our program in the following slides. As I mentioned before, each DCC has its own data model to host their data sets. In CFDE, we have come up with a data model for metadata that helps the users to find and access data easily hosted at different DCCs. This model is called CrossCut metadata model. In short, it's C2M2, and it's a relational database. C2M2's intention is not to capture full depth of metadata available at different DCCs or data generating centers, but to capture enough depth and complexity of DCC's metadata for a researcher to be able to find the data of interest. Okay. Again, I don't want to go off topic, but just want to inform you that data models can be of two different types, or I mean, the relational databases can be two different types. Databases can be in two different types. One is relational database and one is non-relational database. Relational databases store information in tables with rows and columns, while where each row represents a record and each column represents a field. And they are good at maintaining reference integrity and data discovery, data consistency. But there is other kind of database, which is the non-relational database, also called NoSQL database, that are designed to have flexible schemas. They do not use the table. They're not table-based, basically. They're different kinds, and you see some of the examples here. So just to give you context here. All right. So C2M2 is, in its present state, describes various core entities. It describes subject metadata. It describes diseases. It's able to capture biochemical compounds associated with the data, assays, analysis types, and generate the data, I mean, analysis types that are generated by the data. Analysis types that generated the data. Oh God. File compression and various other metadata is captured in various tables. Again, as I said, it's a relational database, so you have different tables that capture different kinds of metadata here. But the core tables are the file table, the biosample table, and the subject table. And we use control vocabularies and ontologies here to capture the full, not the full wealth, but at least a good amount of wealth that is available at other DCCs. And ontologies are really important for data harmonization and also the cross data searches, cross DCC data searches, to be more precise. Okay. So, I just want to go back. So this is a biosample data, a biosample table, and you can see that we are capturing a few bits of metadata here. And I want to just point here that we are capturing anatomy, which is, and we use Uberon for, Uberon is an ontology for anatomical locations. And we use Uberon CV terms for capturing anatomical locations associated with the data. And there is sample prep method for which we use OB. OB is ontology for biomedical investigations, and we use OB CV terms for sample prep methods. Okay. So subject is another code table that I mentioned, and here we are capturing subject metadata. For example, like granularity, sex, ethnicity, and other important information is captured in the subject table. The other important core table is the file table. We captured almost a good amount of metadata about a file, like the size and the checksums, and we capture two different types of checksums, and then the compression format, and if at all there is any kind of persistent ID associated with that particular file, we capture that, and you can see there are other pieces of information in the file table. So as I said, persistent ID is an important metadata. So these are URIs or compact identifiers, which are stable, which means that they do not change. They're kind of permanent. That's why we call them as persistent IDs. And this ID can be used by users or automated in a software, because they're not going to change. So you can use them in automated fashion to capture about the file entity or the file, whatever the file that this particular ID is pointing to. So you can always use persistent IDs to point to that particular file. Okay, so I talked a bit about FAIR in the previous slides, but I did say that these DCCs use different control vocabularies and ontologies for standardizing the metadata at their end. At CFDE, our efforts have been to harmonize the metadata across these DCCs so that the data is interoperable and integrable. So standardization facilitates making data fair. Ontologies and control vocabularies are excellent ways to accomplish standardization. I'm going to talk a little bit about it. Even before going into the details, I just want to briefly explain what control vocabularies and ontologies are. And there is a difference between the two. The control vocabulary is a predefined set of terms. Only terms from the control set may be used, and new terms cannot be made on the fly. Each term has a precise definition. This is understandable by humans and allows them to know how the term should be used. Each term also has a unique identifier. This is a computable object. The ID can be used to tag objects with the term, and the name and definitions are connected to the ID. So these are control vocabularies, but talking about ontologies, ontology is a control vocabulary, but it has relations between the terms. So that is the main difference that between control vocabularies and ontologies. Ontologies have relations between the terms. Each term will have a parent, except the root, and may have child terms, and the children are more specific than their parents. And I'm going to give you a few examples here. Okay. There are hundreds of ontologies out there. If you type ontologies, you will get several examples. And the biggest one, or the most well cited and also well-maintained ontology, is the gene ontology, about with more than 50,000 terms in it. And there are other ontologies for various needs. And like, for example, evidence ontology is ontology with evidence types. Human disease ontology for diseases. And I mentioned about OB, which is ontology for biomedical investigations, for description of experiments, observations, analyses, and there are a few more that OB captures. And also ontology for microbial phenotypes, which is also called ONT, with more than 2,000 terms. So you can find a comprehensive list of ontologies at OBO Foundry. If you go to this site here, you'll be able to see a big list of ontologies for various which cover various domains. And there is also bioportal and ontology lookup service. These are all good resources. So this is a page that shows, this is OBO Foundry page. And you can see that there is a big list of ontologies here that can be used. So before even starting a new ontology, probably you should just go visit OBO Foundry if there is an ontology already existing for the kind of terms that you are looking for. As I mentioned, gene ontology is a well-maintained and most cited, one of the most cited ontologies out there. So gene ontology covers, it is not just one ontology, but it's a set of 3 ontologies. One is molecular function, molecular process, function, and then the cellular component. Here I'm showing a snippet of cellular location ontology where you can see nucleus, which is the term, one of the terms here. It has a proper definition, and it has a parent term, which is intracellular membrane-bound organelle, and then the nucleus is a child of intracellular membrane-bound organelle, which means that the relation between nucleus and its parent is a relation. And there are other child terms under nucleus, and there is also another term called dense nuclear body, which is related to nucleus, not through either relationship, but the relationship between the two is part of. So there are different kinds of relations that you can use between the terms, and that makes ontology different from control vocabularies. So ontologies also will make it very easy for capture of information. So annotation becomes a series of control vocabulary term IDs linked to proteins, genes, and features. So I'm trying to show you an example here. On your left, you can see some text which is taken from a publication, and it's some kind of discovery in the paper, and this text shows some discovery. And how do you want to annotate this? So you can convert this text into annotations, and I'm showing the annotations on the right, and these are all Go annotations. So GO stands for gene ontology, and all the annotations that you see here are what that directly or equivalent of what you see here. So the first one is a process term, and the second one is a function term, and the third one and fourth one are the cellular component terms. So these are the direct annotations of the text that you see here. So and these annotations are computable, which means that you can use them to automate your analysis and to get these annotations and to incorporate in your analysis very easily. And you cannot do that with the text. Okay, and capturing information as logical statements. So for example, I said protein X is a ribokinase that is active in liver cells. And if I have to translate that into annotations, then the protein X can be this uniprot ID, and then has a ribokinase function, which means there is a relation called has function, and then the function Go function term, which is 4747 for ribokinase. And where is this ribokinase or protein X found? It is in liver cells. So you can use Ubron term for liver cells. Ubron 2107 is for liver cells. And where is this annotation coming from? It's coming from a direct assay. So you can use evidence code 00002 for direct assay as direct assay evidence for this particular annotation. And you can see that example here. So this is the power of ontologies. And then just to show the list of ontologies that have been used in CFDE, it's a very comprehensive list of ontologies that are being used in there. And then other thing is that at CFDE, when you find the data of interest, you can take that data to Kavatica. Kavatica is our cloud resource where it has various pipelines and workflows for people to run on. If you have data, as I said, you can take the data to Kavatica and use the workflows and pipelines available in Kavatica to analyze the data there. So all this is made easy at CFDE. So I'm going to quickly show you a presentation here. Raja, do we have some time or are we running short? Just trying to get a feel of, do we have enough time?
Yes, I think you can continue.
Okay, good. So now I'm going to show you a tool called Playbook Workflow Builder, developed by Avi Mayan's lab from Lynx, by partnering with Kids First, Glycosciences, XRNA, and GTEx. And the tool is can be used to build interactive workflows that will show information from across different DCCs using their APIs. So I'm going to click through to show you how this, and here is the web link. You can access this link, and I think this link is also in the important links that Raja showed in the first slide in there. So you can definitely access that link there. So I'm going to proceed here. Okay, so again, this web page shows several cards on the home page. So each card is an entry point for different inputs. You can see that the entry point can be anything. You can go with gene input, or you can go with gene set input. You can go with glycan set input, or disease, or variant, or there are different inputs that you can use to find information about that input in various DCCs. If DCCs have the information available, then all that information will be showing up as a result. So here I'm going to start with gene input. When you click that gene input card, you will see this pop-up box. And in this pop-up box, all that you have to do is enter a gene name. Here I selected Wnt10A, and you then submit. And when you hit submit, you will see another set of cards. And these are the cards that are populated with results from all different DCCs or different resources out there through their API queries. So you can pick any of these cards to know the output or know more information on what that card is giving you. So I'll start with Lynx L1000 reverse search card, and I want to know what results we have there. And when you click that, it will open another window where you can see different perturbation studies, or maybe this is one study that shows different perturbations that are used in that particular study, and how those perturbations have influenced Wnt10A, and also some expression log fold changes here that you are able to see. So this is a nice, so this is coming from an API query with Lynx at Lynx. Now I'm going to click another card, which is query to query GTEx median tissue expression card. And when you click that, it will open another box, and you are able to see GTEx median tissue expression in various tissues and their z-scores. And going back, I'm going to click Kids First gene expression in tumor card, and then you will see gene expression in tumors, various tumors here, and also some other important information from the study, z-scores, and the exact disease or the tumor that is associated with that particular data, and then a few other counts here. So more information about these counts and all the experimental design you would be able to see in the paper. And then I'm going to click glycan protein product page card. And that will open up another box where you can see the glycans associated with Wnt10A and glycosylation sites and some important information, metadata about that particular gene. So here is our publication. And if you're interested in CFDE and want to know more about CFDE, I would suggest reading this paper, and it has all the information and all the links that you can go and explore. And that's the end of my presentation. And thanks to CFDE consortium members, and also thanks to Michelle, who is my boss, for letting me use some of her slides. Thank you. Thanks for listening.